So my dear students, today the topic which I am taking, that is cell division. So let's start cell division. Cell division. So you know that two kind of this divisions we usually study. One is mitosis and one is meiosis. You know that this is our body, and in our body two kind of the cells are present over there. One is somatic cell and one is germ cell or the sex cells. Okay. So in the whole body you see somatic cells are there except the gonads. In the gonads sex cells are there, that is germ cells. So you have to remember one thing, that in the somatic cells mitosis take place and in the germ cells meiosis take place. Okay. So before discussing the mitosis and meiosis, first of all I am going to discuss the cell cycle and the different stages which come in the cell cycle. So let's start. So for example, let's say I am drawing this. Okay, so here in both of the kind of the cell divisions, whether it is mitosis or whether it is meiosis, so here some stages will be there, like G1 stage, S stage, G2 stage, prophase, metaphase, anaphase, telophase, and after that cytokinesis happens. Okay, so let's say this is the cycle I am drawing over here. Okay, and here like this. Okay, so the maximum time the cell takes in the interphase, and the interphase is G1, S and the G2. Okay, so for example this one is G1 stage, this one is S stage, and this one is the G2. So from G1 to G2, from here to here, this time is called as, this stage is called as interphase. Okay, and the maximum time the cell is taking in what? It is taking in interphase. Okay, and the first stage of the interphase is G1, then S and the G2. So this complete one is interphase. Okay, or you can also call it the resting phase. Okay, in this interphase the cell starts the preparation for the division. Okay, so first of all I am taking as the G1, then I will discuss S and then G2. After that, from here to here, let's say this is the next stage which come after the G2, that is prophase. After that metaphase, after that anaphase, and after that telophase. So from here to here, this is called as what, students? This is called as karyokinesis. What is the meaning of karyokinesis? The karyo term is used for the nucleus. The karyo term is used for the nucleus, and karyokinesis means the division of nucleus. Okay, and after that this one is what? This one is cytokinesis. After that what will happen? Cytokinesis means after the division of the nucleus, the division of the cytoplasm will take place, and in this way the division will get completed. Okay, so from here to here, this one I called as interphase, and this phase is what? Karyokinesis, and this one is what? Cytokinesis. And the karyokinesis and cytokinesis is together called as what? This is called as M phase. This is called as what? M phase. M phase is the phase of actual cell division. The actual cell division is taking place in this stage. Okay, here in interphase the cell is just doing the preparation of the division, right? So in M phase I can say this is the phase of actual cell division. Actual cell division. The actual cell division is taking place in the M phase. Okay, now in your NCERT one thing is written over there, that usually karyokinesis is followed by the cytokinesis. It is writing usually. Why the NCERT is using the term usually? Because after karyokinesis cytokinesis happen, it's not necessary. Okay, so in M phase, this phase will be the phase of actual cell division, and usually starts with karyokinesis, and followed by cytokinesis, Okay, for example, but one exception over here, but every time the cytokinesis will not happen after karyokinesis. One exception you have to remember over here, that the coconut endosperm, in case of the coconut endosperm, the cytokinesis is not happening after karyokinesis. That's why the multinucleated condition is developed in the endosperm, and the coconut endosperm, that multinucleated condition is called as syncytium. This is called as syncytium. Okay, so this is actually the actual phase of division, right? Now I talk about the interphase, and interphase, first of all I am going to discuss what? G1 phase. G1 means what? It is the first growth phase. This is the first growth, or you can call gap phase, first growth or gap phase. Okay, this one is what? This is what? Most complex. This is what? Most complex stage, and the cell in this stage can be at for the variable time period, so I am also calling it that it is variable, and this stage is also called as deciding phase. This is also called as deciding phase. Why we are calling it as deciding phase? Because the cell has to decide in the G1 phase only that whether the cell will go for further stages like G2 prophase, metaphase, anaphase, telophase and cytokinesis, whether the cell will divide or not. All the kind of the decisions are taken in the G1 phase. That's why we are calling it what? Deciding phase. Okay. Now this G1 phase is having multiple functions over here, like ATP is synthesized, nucleotides are synthesized, yes or no? Cell organelles number is also increased over here, yes or no? And in the G1 stage, sometimes what happens that some cells leave the G1 phase. Okay, in the G1 phase ending, the cell has to decide whether it will go for the division or not. But some exceptions over here, that some cells go out of the G1 phase, they enter into the G0 phase. Okay, either they can enter into the G0 phase, it is a kind of inactive stage. Either it can enter for the temporary way, or it can go the permanent also. So if the cell is going into the G0, it means that it is going out of the cell cycle. It may be the temporary. It means that when the nutrients are available, that it can come back to the cell cycle and further it can follow the cell division cycle. Okay, but in some cases what happened that after G1, in the G1, some cells go into the G0 stage permanently. Okay, and do you know the examples of this? So examples you can remember that in case of the neurons and in case of the heart cells and the neurons, they do not come back into the cell cycle. It means they do not divide. So every decision is taken into the G1 phase. Okay, after the G1 phase, the cell has to enter into the S phase. But here between G1 and S, one checkpoint is present over here. This is called as checkpoint number 1. This is checkpoint number 1. This is checkpoint number 1, or you can also call it restriction site. Restriction site. Okay. For example, when we are going to the city, and in the way you see the traffic over there, and at the some places the policemen are there, and there's a checking that what is there in your bike or in your car. Okay, so these are the checkpoints. Like that way, the cell also has the checkpoints, so that it can check that whether the cell organelle number has increased properly and all the preparations are well or not. So all these kind of the preparation to be checked at the checkpoint, that is the checkpoint number 1, that is also called as the restriction site. After that, the cell enter into the next stage, that is called as S stage. This S stage is called as synthesis phase. This is also called as what? Synthesis phase. Synthesis phase. Okay, this is also called as trigger phase. Also called as trigger phase. Why we are calling it the trigger phase? You know, trigger, gun, trigger, like that. So you understand one thing, that in S phase something special happens. That in the S stage what happens here? DNA replication happens. DNA replication. DNA make its copy. DNA replication. Or what other thing happen, that centriole duplication happen in this stage. Centriole duplication. Centriole get doubled and DNA make its copies. Okay, in what stage? S stage. You understand one thing, if the cell has replicated its DNA, then the cell must have to divide, and if the cell is not dividing, then the what will happen, that particular cell will be destroyed by the apoptosis method. Okay, so if the cell enters into the S phase, then the cell must divide, because here the DNA replication has taken place, centriole duplication has taken place, and this phase named synthesis phase or trigger phase you have to remember. Okay, So DNA replication happens. So if the DNA replication is happening into the S stage, then definitely what will happen, that the DNA amount will get doubled and the chromosome number will remain same. Now here one confusion comes, that sir, if chromosome number getting double, if the DNA is getting double, then you are saying that chromosome number will be same. How it happens? So students, focus here, that for example, let's say this is the chromosome, this is the chromosome, okay, and if I say that what is this, then you will say, sir, this is also chromosome, right? If I ask you one thing, that students, how many chromosomes here, then you will say, sir, here one chromosome is present over here, right? And if I ask you here how many chromosomes, you will say, sir, here 2 chromosomes is there, but that is wrong. Here 2 chromosomes are not there. Here is also 1 chromosome. Here is also 1 chromosome. Then what is the difference? Then here 1 chromosome, students, 1 chromosome with 1 chromatid, and here 1 chromosome with how many chromatids? 2 chromatids. For example, let's say I am, right? If I am having 1 hand and 1 leg, then you will say that how many persons are there? Only 1. That 1 person with 1 hand and 1 leg, right? But if I am saying that now how many persons, then you will say, sir, now also the person is 1 only, but now the person is having 2 hands and 2 legs, right? In the same manner, that for example, this is the chromosome, this is 1 chromosome, and this is also 1 chromosome, this is 1 chromosome with 1 chromatid, and this is 1 chromosome with 2 chromatids, okay? So what is actually happening in the S stage, that here the chromatin which is present here, this chromatin is making its copy. This chromatin is making its copy, right? Like that. DNA replication is happening, so it is making its copy. So you understand one thing, here also 1 chromosome, here also 1 chromosome. This is 1 chromosome with 1 chromatid, 2 chromatids. But if I talk about the DNA value, let's say here the DNA value is 2C. Okay, and here the DNA value will be what? It will be 4C, but still the chromosome number is what? Same. 1 chromosome, but the DNA has been doubled, 2C to 4C, because 1 chromatin value has become the double. So 2 chromatin value has become the 4 chromatin value. So what is happening between G1 stage and the S stage? G1 stage and the G2 stage, S stage is coming in between. So let's say this is the G1 stage and this is the G2 stage. In between the stage, that is coming the S stage, and in the S stage the DNA making its copies, right? That's why we say that between from G1 to G2, in between the S is coming, DNA is getting double, but chromosome number is what? Chromosome number is same, right? The next come to the G2 phase, and the G2 phase is called as what? This is growth second phase. Growth second phase, or you can say gap 2 phase also, right? So this is the G2 phase. In the G2 phase, some tubulin proteins will be formed because central duplication has taken place in the S phase. So the tubulin proteins will be formed, because what will happen when we'll go further? So you will see that in the metaphase chromosomes are visible, right? In the metaphase, you can see the chromosomes properly, and the spindle fibers will be attached over there, right? So for that attachment, tubulin proteins are formed into the G2 phase. So this is growth second phase, right? After that, what will happen next? Prophase will come, metaphase, anaphase and telophase, and finally when the cytokinesis will happen, the cell will get divided, and in this way the cell division will get completed. Okay, thank you. We'll meet in the next class. You have understood one thing clearly over here, that between S and the G2 in the S phase, between G1 and G2 in the S phase, DNA replication happened, central duplication happened. Due to DNA replication, the chromosome number is same, but due to DNA replication, the DNA amount has been doubled, but the chromosome number is still same. Why the chromosome number is same? You understood this concept very well. Okay, we'll meet in the next class with a new topic. Thank you very much. See you.